Start the session. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh.
The Honorable, the Head of Department of Pharmacy, Universitas Islam Bandung, Mr. Dr. Swender, along with his staffs. The Honorable, our invited speaker, Professor Noriyuki Koibuchi, M.D., Ph.D., and respectable, the lecturers, researchers, and all participants of International Sharing Session 2021. Alhamdulillahirabbil alamin wassalatu wassalamu ala asyrafil anbiya'i wal mursalin sayyidina Muhammadin wa ala alihi wa ashabihi ajmain amma ba'du. First of all, let us praise and thank the presence of Allah Subhanahu wa Ta'ala for the abundance of grace and joy so that we can still gather in this virtual event in a good health. Secondly, the salawat and greetings we pour out to the Prophet Muhammad Sallallahu Alaihi Wasallam. May we all get intersection on the end. Amin ya rabbal alamin. Ladies and gentlemen, welcome to International Sharing Session 2021 that organized by Department of Pharmacy Universitas Islam Bandung. Today, we invited an impressive professor from Department of Integrative Physiology, Gunma University Graduate School of Medicine, Professor Noriyuki Koibuchi, M.D., Ph.D., to talk about endocrine disrupting chemicals, from basic research to its implication. Maybe some of you is already familiar with EDC, but we will talk more deeply today. So endocrine disrupting chemicals or EDCs are substances in the environment. It could be from air, soil or water supply, food sources, personal care products, and manufactured products that interfere with the normal function of our body's endocrine system. Some EDCs act like hormone mimics and trick our body into thinking that they are hormones, while other EDCs block natural hormones from doing their job. EDCs may increase or decrease the levels of hormones in our blood by affecting how they are made, broken down, or stored in our body. So EDCs can disrupt many different hormones, which is why they have been linked to numerous adverse human health outcomes, including altered nervous system function, immune function, growth, neurological and learning disabilities, and many more. So to know deeply about EDCs related hormone and brain development, we will hear the explanation from Professor Noriyuki Koibuchi. So ladies and gentlemen, before we start this session, let me introduce Professor Noriyuki Koibuchi by saying his curriculum vitae. All right. So Professor Noriyuki Koibuchi is a professor and director of Department of Integrative Physiology, Gunma University Graduate School of Medicine. It's in Maebashi City in Gunma Prefecture in Japan. So he started his medical degree in 1985 in Gunma University School of Medicine and continued his PhD also in Gunma University. And after he finished his PhD, he continued the postdoctoral training, and the latest one in the Rockefeller University in New York. And he's now the registered physician, Japan Diabetes Association. And if I read all your curriculum vitae and say maybe it spent more than an hour. so right now his position I already mentioned before. And then, maybe people need to know what's your major research interest. So his interest is in molecular mechanism of thyroid hormone action on organ function and development, and then in molecular mechanism or orphan nuclear receptor action, the effect of environmental chemicals on nuclear receptor-mediated transcriptional regulation, and also about the tropic regulation of brain development and the influences of environmental factors on its process. So in bibliography, of course, Professor Koibuchi wrote so many books, and the latest one is, right, okay, on 2018 about the molecular mechanism of thyroid hormone synthesis and secretion. So it's, is it available in Indonesia, Sensei? I don't know.
I don't know.
Maybe later we can order if you're interested to. And then, of course, he wrote a lot of reviews, articles, and original articles because he has a lot of projects and then students, of course, and colleagues. And maybe I'm one of them in here, right? At least 3 articles maybe
Yeah, I can
we
see here, number 31.
number 30 and 31. Okay, and a lot of original articles. So
Mm.
he's has so much experience in writing articles, of course, and because he's also editors of some of journals. So if you guys need some suggestion from Koibuchi Sensei of Professor Koibuchi now, you can try to explain your proposal here orally, right, Sensei? And
Mm.
maybe Koibuchi Sensei will give you any suggestion. So let's start this session. Please welcome Professor Koibuchi Noriyuki Koibuchi, please. Sorry.
Okay, thank you very much, Miski. Uh, Selamat pagi, ladies and gentlemen. It's my great pleasure to share the time with you. Actually, I wish I could visit Indonesia, but unfortunately, because of the COVID-19 tragedy, I cannot visit you. I love Bandung. I have visited 5, 6 times, you know. So I wish next time I could see you face to face. Okay, so I have heard that some of you may not speak or understand English very well, so I will try to speak slowly. But if you think that I'm speak too fast, please let me know. I'll try to speak slower. Okay, I will share my slide.
Sensei,
Yes,
excuse me, please, before you start your session.
yep.
Ladies and gentlemen, please, if you can open your feature camera and we can see you all here. And if you have any question, even you write in Indonesia, it's okay. Please directly write your question in the chat feature. Thank you. Gozo Sensei. Sorry.
Okay, thank you. All right, again, I will share my slide. Here you go. Okay, so before I talk about science, I would like to introduce a little bit about Gunma University. Okay, so Gunma is located just in the middle of the Japan, which is about 100 kilometers north from Tokyo. So some people say Gunma is kind of very bottom of Japan. So we have a lot of national park here. I believe that Miski have been some of these. Um, we have very beautiful mountain, and you can see the very nice foliage in autumn. And we have a very nice highland area, and you can enjoy skiing in winter. And also we have a very famous place for the hot spring. And of course, sometimes you have to be naked, but recently you're allowed to wear the bathing suit in hot spring, so you need not worry too much. And we also have a lot of festivals. This is Obon dance. This is to celebrate our ancestors' spirit in summer. This dance is usually accompanied with fireworks like this, and we have very nice firework in Maebashi. Also, we have some parade wearing the samurai costume. I believe that Miski also have been there to see. It is in Numata, right? You have been
Yes,
there.
yeah.
Okay.
You know well.
Yeah. And this is a Samba festival because we have a very close relationship with the South American people, and we have a lot of immigrants from South America. So we also have a Samba festival in Gunma. And this is the Gunma University Graduate School of Medicine, which is located in the capital city of Gunma, Maebashi. And we have founded in 1943 as a Maebashi Medical College. And then it became the Goomba University School of Medicine in 1947, and then which was transformed to the Graduate School of Medicine at 2003, meaning that our university is a research-oriented university, not only to educate students to be a doctor, but also we are to educate students to be a scientist like Miski.
Really.
Okay, so this is the main gate of Gunma University. I believe that Miski means the cherry blossoms in Gunma University. In spring, we have very nice cherry blossoms.
Mhm.
And this entrance to the university hospital. and the inpatient facility. And we have a lot of imaging tools such as PET, SPECT, MRI. And we have a heavy metal ion therapy center. Only 3 universities in world had this heavy metal ion therapy center. And this is very useful because you need not give the surgery for the cancer patient. You can just give them heavy metal ion, and then you can mimic the surgery. And we have a skill lab training center for MD and pharmacist and nurse or whatever. We have more than 40 research laboratories, and we have more than 500 publications per year, including Nature, Science, Lancet, JBC, Cancer, Share, Blood, Endocrinology, etc. And this is the our laboratory member. You can see Miski here. We also have, we used to have other Indonesian named Winda. She's also from Bandung. She used to work in the Bandung Institute of Technology. And also we have another Bandungese, Arfan. He is from University Pajajaran. We also have one Malaysian, one Syrian, and she looks like Japanese, but actually she's from Australia. So she did very great in Japan. Miski did very great in Japan. Of course, she's always cheerful and funny. So sometimes she enjoys herself by dance, showing the traditional dance from the Indonesia. You remember this, right?
Right, yeah, I was pregnant actually.
Yeah, the Christmas party. Yeah. Oh, you were pregnant at the moment?
Yeah.
Okay, I understand. That's why you're buried larger. Not because you're fatty.
No, it's okay.
Okay. Anyway, so I love watching her dance, but also she did very great job in science. She has 2 very good, very nice article in the very high top level research paper journal. One is in investigative radiology. The impact factor of investigative radiology is about 7, which is very high. And she's the first author of the this paper, and she talked about the EDC, environmental some chemicals named gadolinium. I'm going to talk a little bit more about this topic later on. Also, she published other paper in Frontiers in Endocrinology, which is also the top journal in the field of endocrinology. And she used the propylthiouracil. This is a medicine used for the basidosis patient, our Graves disease patient, and she also did a very nice job in this research. Anyway, and I would like to introduce myself. My name is Noriki Koibuchi. I was born in Maebashi, Gunma, and grew up Hitachi, Ibaraki. Probably you know the name Hitachi because this is a very famous place for the electronic company. And then Hitachi was born in this city. And right now, I'm a professor and director of the Department of Integrative Physiology, Goomba University Graduate School of Medicine. And my research specialty is endocrine physiology and environmental physiology, including EDC research. And also, I'm working as a clinician as well. And my specialty, endocrinology and metabolism. And I'm registered diabetologist in Japan. So actually, I used to work in Harvard Medical School, and I sometimes take my student to the Harvard and do the clinical practice. Okay, so as Miski mentioned before, we will talk about endocrine disrupting chemicals, particularly on neurodevelopment, because brain is the primary target of EDC. And the exposure of EDC during fetal period or early postnatal period cause a lot of abnormalities in brain development. Okay, so at first, some of you may not know very well about the developing brain. So I talk a little bit about the characteristics in developing brain. And also I'll talk a little bit about hormone and brain development. And then I'll talk about the mechanism of lipophilic hormone action. And also I talk about, of course, EDC and the action in the developing brain. Then I'd like to invite all of you to discussion session. Today's your task is not to just hear my talk,
Right.
but in the half 60 minutes, first 60 minutes I'll talk, but next 60 minutes you need to discuss or you need to talk to me. Okay. So this is very important. You need to design your experiment by yourself after listening to my talk. And probably on the way of my talk, Miski will provide handout for you to discuss more, right, Miski?
Yes.
Okay. Okay, I'll talk first about the important characteristic and the brain in developing brain. So, brain is not like other organ. Gene expression in brain is temporally and spatially specific. That means development of brain occur in time-specific manner. For example, cerebral cortex develop first, but cerebellum develop last. Cerebellum keeps developing even after birth. Okay, so and each development process is controlled by different gene. So neuronal proliferation is controlled by certain gene, gene A, and neuronal migration is controlled by gene B, and dendrinogenesis is controlled by different gene C, something like that. So in each development period, different kind of gene is expressed in the brain. So all neurons go through this process. At first, proliferation, migration, and dendrite axon growth and synaptogenesis. So a distinct critical period exists in each brain region. Okay, in some brain region, cerebral cortex, for example, proliferation starts immediately after fertilization, but in cerebellum, proliferation goes on even after birth. So disruption of gene expression at specific development period affect specific neuronal event. Everybody understand what I'm talking about?
Yeah, you can continue. I think.
Okay. So, for example, neurons start to proliferate, then it migrate to their final destination with differentiation. Then axons try to elongate by using their roadmap, some of which could be neurons, some of could be plus or minus charge, or some of which could be chemicals. So axons start to elongate. At the same time, dendrite start to grow, and synapse can be formed between dendrite and axon. So, for example, in cerebellum, Purkinje cell differentiation start at embryonic stage E50. This is a rat and mouse. So Purkinje cell differentiation start at here, but the granule cell, this is a different subset of cells in cerebellum, develop after birth. P means postnatal, E means embryonic. Okay. So for the healthy brain development, influence from the environment plays very important role. I'll talk more in detail. So optimal environmental stimulation is necessary for normal brain development. And brain function is controlled by neuronal network, not by single neuron, okay? So each function is controlled by network, not single neuron. That means abnormal gene expression or disruption of gene expression in a specific subset of neuron may not be detected in childhood. For example, only one class of neuron is disrupted during development, you may not be able to detect such abnormality, because neuronal network develop, throughout, postnatally and prenatally. Okay, so again, at first, brain cells start to develop by using intracellular programming. However, you have to have sensory stimulation to transmit environmental information. For example, after birth, when you cover, immediate after birth, when you cover your eye like this, then remove your cover 1 year after birth, then your right eye cannot see anymore. Okay, so light stimulation is necessary to develop visual information. Understand? So also in ear, if you cover your ear for like 2-3 years after birth, then remove it, then you may not be able to hear anymore. So sensory stimulation is very important for normal development. Also, hormonal stimulation played a very important role in developing brain development. For example, when you're a child, your mother may yell you if you do badly, right? Then glucocorticoid or steroid hormone could be secreted from adrenal cortex. Such glucocorticoid can protect your brain from brain damage by yelling. So hormones such as estrogen, or sex hormones, thyroid hormone, or glucocorticoid play a very important role for normal brain development. However, very strong stress or environmental chemicals like EDC or starvation may disrupt this sensory stimulation or hormonal stimulation. Okay, this is a very big problem. So very strong stress can induce a very high level of glucocorticoid, which may kill the brain. Or sensory stimulation can be very strong by strong stress or strong pain or heat or something like that, which damage the brain cells or damage the brain programming. Of course, environmental chemicals, this easily disrupt this environment, and starvation also may disrupt the brain development. Okay, so why endocrine disrupting chemicals easily disrupt hormones? Okay, to understand, I'll talk a little bit more about hormones. Hormone has two distinct actions. One is, I would say, organizational effect. So again, organ morphogenesis and functional differentiation is controlled by hormones. But again, as I told you, different hormones regulate different neuronal event. So each hormone have distinct critical period. Okay. So for example, you may have heard about cretinism or maybe iodine deficiency. So iodine, if you find iodine deficiency during pregnancy or immediately after birth, you need to start replacing iodine to inhibit thyroid hormone deficiency. Even if you start replacing iodine when child becomes 3 years old, you can't reverse abnormal brain development anymore because thyroid hormone action on brain development is only effective only during the critical period, which will be younger than 1 year old for the kid. Okay, so each hormone has such organizational effect. So, but a small abnormality may not be identified at neural period. For example, ADHD, for example, attention deficit hyperactive disorder can be induced by little deficiency, small deficiency of thyroid hormone. You know, I would like to show Miski data later on, but you know, it is very difficult to find out. And gender identity also, you may not be able to find until such kind of identity disorder until puberty. But abnormality start when you're in fetus, okay? And higher brain dysfunction, also memory or motor balance or something like that. On the other hand, hormone also have activational effect. This is to regulate homeostasis, such as fluid volume, glucose metabolism, such as insulin, and menstrual cycle after puberty. These hormones may also have this kind of activational effect. This effect persists throughout their life. But again, this organization effect persists only during the critical period. Understand? So the problem for EDC, environmental chemical, is if you are exposed to environmental chemicals, EDC, at certain period during pregnancy or in lactation period, some neuronal event could be disrupted by inhibiting these hormone actions. Okay? So many hormones affect in the brain, but I will show you the several representative hormones. One is sex hormones, such as estrogens, androgens, and of course, these are responsible for gender differentiation in like a penis or vagina or breast or muscles or voice, but still also it regulates brain sexual differentiation. So believe or not, male brain and female brain is different structurally and functionally. Okay, these sex hormones regulate brain sexual differentiation. And glucocorticoid, as I told you, glucocorticoid regulates stress tolerance. So certain level of glucocorticoid is important to construct stress tolerance. And neurosteroid, recent studies have shown that many steroid hormones are locally produced in the brain, produced in the brain, but still function is very well known. Antithyroid hormones, my favorite hormone, is regulate neuronal development. Particularly intellectual development is regulated by a thyroid hormone. So why hormone action is easily disrupted? Because plasma concentration is very, very low. For example, thyroid hormone level in plasma is 0.8 to 1.8 nanogram per liter. You know, this is 1 trillion, 1 part per 1 trillion. Okay, probably you have heard ppm, right? For example, if someone express air pollution, you may have heard that oxidant level is 1 ppm. Is it familiar also in Indonesia?
Yeah,
Yes, ppm.
Right. So ppm is familiar. Yeah, we may consider that 1 ppm could be very low, right? Because 1 part per 1 million. However, hormone level in plasma is much less than ppm. I would say part per trillion, so ppt. So 1 drop of EDC in, how to say, one bucket, one large bucket, or one bathtub, one drop of EDC may disrupt hormone action. You understand? Right? Very low level. Also in estradiol level is a little bit higher, particularly during ovulation. It's 300 nanogram. But again, it is still like a 10 to 300 per trillion. So estradiol level is still very low. And testosterone level on male will be a little bit higher, but still 2.5 to 10 per billion. So thyroid hormone level, estradiol level, or other hormone level also, plasma concentration is very low. So only if you drop of endocrine disrupting chemical can easily disrupt our hormone environment. Okay, and some hormone may act through membrane receptor. Other hormones may act through nuclear receptors, but many hormones affecting in brain can act through nuclear hormone receptors. So hormone can pass through the membrane. Sometimes you have to have transporter. For example, thyroid hormone requires transporter to enter into the cell. Steroid hormone may not necessary to use transporter. Anyway, hormone can enter into the cells, which can then bind to the nuclear receptor. Then these nuclear receptors can bind to hormone response element of the target gene. Then usually messenger RNA transcription is accelerated. Then S is missing. Then messenger RNA can be translated to protein, and this protein has various actions. So disruption of hormones may disrupt the expression of proteins. Okay, sorry, it's in Japanese. This is advertisement, enrich yourself by chemical substances. I don't think it's true though. Okay, so EDC is endocrine disrupting chemicals of an exogenous substance or mixture that alter function of the endocrine system and consequently causes adverse health effect in an intact organism or its progeny or populations. This is defined by WHO in 2002. So again, since plasma concentration of hormones are very low, ingestion of such chemicals may disrupt our hormone environment even at a low dose. And also, half-life is very long because it is, thinking about our evolution, it is quite recently when we start to be exposed to this kind of endocrine disruptive chemicals. Okay, it was like maybe 100 or 200 years since we are started to be exposed by this kind of chemicals. So you do not have system to catalyze well of these substances or to excrete well to these substances. So we have so many endocrine disrupting chemicals harboring hormone-like actions like herbicide, this is insect killer. These you need not to remember, and this is too many. And fungicide, this is also used for the pharmacy in farm. Insecticide also. We have so many chemicals and heavy metals, organic tins, mercury, cadmium, gadolinium, lead, manganese, So these, I don't know why, but these heavy metals can be also disrupt hormone environment. And polyphenols, isoflavone, we eat a lot of isoflavone because we eat a lot of beans and catechin. We drink a lot of tea, so these can be also disrupt hormone environment. But fortunately, these have actually health promoting effect. Actually, this is more profitable than disruptable, fortunately. And industrial chemical products, bisphenol A, polycarbonate, butyl hydroxyl or bromophenol or dioxins. Probably have heard about the dioxin, right? Probably it's pretty famous EDCs. And probably you have heard of PCB. This is also representative endocrine disrupting chemicals. And of course, we have uh, other prescribed drugs, which is also unfortunately secreted into environment. Hormone agonist, amiodarone, because your background is a pharmacy, many of your background is a pharmacy, also I believe that you know the name amiodarone, or dative this is a synthetic estrogen, phenamic acid, this is anti-inflammatory drug, but this is also have the hormone disrupting action. Phenobarbital, this anesthetics, phenytoin. And also we're gonna talk a little bit about gadolinium today. This is also used for diagnostic purpose, but this is also secreted into urine and polluted in river. And this become very big problem in Japan because many prescribed drugs are secreted into urine and pollute the river and the lake and of course ocean. So these are also considered the endocrine disrupting chemicals. And so far, human being invented more than 100,000 chemicals, but possible chemicals having endocrine disrupting action is, I would say, 1,000. But this does not mean 1,000, only 1,000 chemicals have EDC action. We cannot, sorry for my typo error, mostly unclassified due to no published data. And you will see how difficult it is to identify toxicity of each chemicals. You have to have a lot of many different kinds of experiments to identify. And each organ have different level of toxicity. Each organ have different function, action of EDC. So it is very, very difficult to identify. So far, roughly 1,000 chemicals has been studied to have EDC action. Okay, sorry, something. So multiple site of EDC action. For example, EDC may inhibit the differentiation or proliferation of endocrine organs such as thyroid, or inhibit secretion of hormone, or inhibit binding of carrier protein in serum, or inhibit transport to brain, or inhibit reduced hormone sensitivity in brain, or EDC may act to other organ, which may indirectly act brain development, or EDC may have direct actions to other organs, or EDC may also inhibit the metabolism of these hormones. So how can we assess the toxicity of these hormones? You can easily understand that it is very, very difficult to examine the effect of EDCs. But usually we focus mainly on hormone sensitivity because many EDCs have direct action to hormone receptors or brain. So we usually focus mainly on this system anyway. So I would like to show one typical example, which is PCB. This stands for polychlorinated biphenyl, which have 209 conjugates due to different chlorination. And this chemical nature is very similar to oil, but inflammable. And actually, if you compare the structure of PCB and dioxin, it's very similar. PCB can bind, benzene ring of PCB bind together directly, but dioxin, in case of dioxin, benzene ring is bound using dioxybound. And when you compare these to thyroid hormone, you can easily identify the chemical structure is very similar, right? All of these have two benzene rings. These benzene rings are connected with halogens, right? In case of PCB and dioxin chloride, in case of thermal, it's iodine. So by comparing the chemical structure, you can easily identify that these could disrupt thyroid hormone action as it does. So again, PCB has been widely used in industrial household appliances and electric fluid transformer and capacitor, like these, and hydraulic lubricant paints, copy paper, etc. So even now, even in Japan, PCB is still used because once this transformer is banned, then these could be damaged in the store site. PCB could be flow out, and it may pollute the soil. So still we keep using this kind of transformer. I believe also in Indonesia, right? Have you heard about it? Yeah.
Yeah.
So still a lot of PCB contaminated transformer is used in in all of the world. But production has been banned in 1972. Oops. Approximately 1.5 million tons has been produced. And most PCB still globally exist in environment because of its chemical stability. Okay. And again, use of PCB content appliances still allowed. Nano and picomolar of PCB is contained in blood, breast milk, and almost all natural products. Okay. So, what's going on? Sorry, what's going on in here? Is this my side? Just wait. Something seems too wrong. Okay. Okay.
Is it fine? Okay, good.
Okay. Yes.
Yeah.
You get it? Can you see?
Yes, sure.
Okay.
Yeah.
Okay. Okay. So this is a very famous effect of PCB. So many PCB was produced around Michigan Lake region in the United States. So the waste PCB is dumped in Michigan Lake, and the fish eats PCB contained water, and because PCB cannot be catalyzed in body, so the fish was contaminated by PCB. So a mother who ate fish took in Michigan Lake were exposed to PCB, and these mothers had a baby, and so their babies are also exposed to PCB during fetal period through placenta and through breast milk. So people measure the PCB concentration in maternal milk. So that with the increase in PCB concentration in maternal milk, ID of the newborn decreased. And sorry, there is no error bar, but you can see the clear tendency again. Reading mastery and word comprehension can be also decreased with the increase in the PCB concentration in maternal milk. So the more fish they took, the higher PCB concentration was seen. Okay, and I do not want to show the result of our experiment, but we found that this PCB effect is exerted by inhibiting thyroid hormone action. Okay, so thyroid hormone action is during development, is disrupted by PCB and brain development was disrupted. Okay, so up to here, do you have any questions?
So since I previously there's questions from Mr. Rinaldi Tanda, maybe it's in the slide page 4, I guess. It's about the
Obesity. Okay, this one, right? What is your opinion about possibility? Oh, is this
sorry,
one?
the previous one there's another questions. I
Okay.
mean in this page 4, could you see back? Yeah, he asked about the starvation on your previous slide that written on page 4.
Starvation.
Yeah, what that it means, he asked.
Oh, sorry.
More than that. It's page 4, I guess. I don't know. It's in the handout. It's
This is next one, right?
yeah,
Here.
that's one. So here, yeah.
Starvation.
Yeah, it
Yes.
means hunger.
Yes, hunger.
Mm,
You mean like this is pretty famous. Like I would say malnutrition. Sorry, starvation may not be the right word. Malnutrition. If nutrition status of the baby is not so good, then brain development disrupted because brain cannot receive enough amount of nutrition, right, from environment. That's causing the abnormal brain development. Okay.
All right, and maybe it's enough, Mr. Rinaldi. Is it understood?
Yes, it makes more possible if it mentioned to malnutrition, not starvation.
Sorry.
Because
Yeah.
yes, because everybody can get starving, and
Right.
I
Ramadan,
think
right? Well, I have one comment about Ramadan. Actually, I have one paper. I have one publication regarding Ramadan
okay.
with one of my Indonesian colleague. Time restricted nutrition is very good for your health.
Oh.
You know, you need not control the amount of food that you took, but you can restrict the time of eating. Then you can inhibit yourself from metabolic syndrome. Okay, obesity can be inhibited by time restricted feeding. I have such paper. Yeah. Okay.
Okay, thank you, Prof.
Thank you. Okay, and another question from Mr. Yuda from Stikes Natakusomo. He asked, what is your opinion about possibility that EDC could contribute to development of obesity, especially in estrogenic hormone? Thank you.
Yes, this is very famous. Somebody believed that increase in the population obesity can be part of which can be due to the ingestion of environmental chemicals. Not only estrogenic or glucocorticoid or how to say thyroid, EDC may also affect or also may disrupt, let's say, leptin action or other home action related to the food intake. So because of these reasons, EDC may influence the obesity. Okay, is that enough? Yeah.
Alright. Okay. Thank you, Prof. And the next question from Ms. Esti Rahmawati. Thank you very much for a very interesting presentation, Prof. Koibuchi. I would like to ask, is there any specific brain area as the target of EDC or hormonal changes, or it will affect all brain areas?
Everywhere.
It's my study.
Everywhere.
Everywhere.
Everywhere. But again, the sensitive period is different. Okay. Many endocrine disrupting chemicals may affect, I would say, synaptogenesis rather than differentiation, because like differentiation of the cerebral cortex is disrupted by large amount of EDC, then fetus cannot survive, right? But very little amount of EDC can inhibit synaptogenesis or dendritogenesis. People can survive, right? So in many cases, synaptogenesis is mainly disrupted by EDCs. So again, timing of synaptogenesis differs depending on the brain region. So depending on the time of exposure, different area of brain could be affected. Is that is it okay to? Yeah.
Miss Esti, would you directly open your mic to ask?
Yes, thank you for your explanation, Professor. I would like to ask more, how about the adult brain? Is there any effect from the EDC to the adult's brain, especially in synaptogenesis? Thank you.
Well, I believe yes. I believe yes. Probably synaptogenesis in adult could be also affected. But again, compared with the synaptogenesis in newborns and the adult, synaptogenesis in newborn or fetus will be more active. So the influence will be more severe. But of course, EDC could be related to the etiology of the Alzheimer's disease or Parkinson disease or other dementia-related disease. But still, we need to work more to clarify the mechanism. But I would say likely. Okay.
Okay, Miss Esti, is it quite understood for the explanation? So we can move to another person. So Miss Siti Nadira wants to ask to you directly.
Okay.
Please, Miss Siti Nadira. Could you unmute your? Okay.
Yes.
Okay, sure.
Hello, Prof. Koibuchi.
Hi.
I hear Japan is discipline country. I want to ask you how to make our brain can think positive. Sometimes if we think positive, enemy can stay from behind and beside.
Sorry. I do not understand very well.
Could you please repeat?
I want to ask you how bagaimana agar kita selalu bisa berpikir positif gitu, Miss. Sometimes if we think positive, terkadang gitu, kadang musuh dari bisa menusuk kita dari depan dan dari belakang juga gitu, Miss.
I see. It's kind of like not really, how to say, related to your presentation, but maybe it's the stimulus of the positive thinking of our way. Like you know, like
Okay.
how we can think positively, then is it affect our brain or our behavior, then how it works, you know, like in Japan maybe kind of the environment is really health, I guess, I don't know, yeah.
Well,
Trying.
it's very difficult question. Positive thing. Actually, positive thinking is very important for the brain to grow healthy. But again, little stress is also important
Mhm.
to help your brain grow healthy.
Oh really?
Yes, again, as I told you, like recently, we have a problem, for example,
Mhm.
like some mother takes care of the baby without yelling at all.
Mm.
They just say, "Oh, you are a good boy. You are a good boy. You are very nice. You behave always very nice."
Mhm.
Then, when they grow, when they become the university student, for example,
Mhm.
then sometimes they make mistake. Sometimes I try to yeah, or sometimes I use very strong word
Mm.
to them in order to make them to be a good doctor.
Mm.
But some students become very depressive by hearing such kind of negative words because this is their first time to listen such kind of strong words from teacher or others. So such student can be very much depressed, can be easily depressive. So of course, positive thinking or good care is important, but little stress is also play very important role for your brain to grow. Is that is that can I answer correctly for your question?
Sudah terjawab mungkin Teh Siti jawabannya. Ya, jadi kalau
Terjawab.
kalau kalau stres sedikit-sedikit bagus ya. Supaya otaknya berkembang. So little stress may help the brain grow up, yeah. But I don't know if the too much stress is kind of
Right.
okay. Sensei, maybe that's the questions I've read in the chats feature. Would you continue your discussion session?
Yeah, we have another part
Mm-hmm.
of discussion, and I would like to let attendees think.
Mm-hmm.
Okay,
Mm-hmm.
so we can talk a little bit more, and I also have more realistic data. So
Mm-hmm.
you may have some additional question after this session. So can I continue?
Yeah, sure. Yeah.
Okay. Okay. Oops. Okay, so we're gonna move on to discussion session now. So, as I told you, assessment of EDC toxicity is very difficult, right? But we should assess the toxicity. So, again, it takes time until phenotype appears because, again, you may not realize your child's abnormality until they grow a little bit older to express their feeling or their intellectuality, but still you have to study the toxicity to avoid
Mhm.
being intoxicated. Okay, so in human study, other factors may be involved. For example, you may consider this is very clear, right? With the increase in PCB concentration, maternal milk, IQ could be disrupted. But so in other words, with an increase in fish intake taken from the lake, for IQ or reading mastery could be decreased. But even with this data, some researcher does not believe that it was caused by PCB. Why do you think so? Why some researcher does not believe? Can you think about it? You can just write down the answer in chat.
Yeah. Why?
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 This is a really difficult question.
So
But
is the lowest one is the concentration in the lake or in the plasma or some
plasma,
methanol milk.
milk.
I see.
What do you think? I have a very big discussion with some research actually when I show this data in the International Symposium.
Okay. Anybody? Is there any comment from me? Is it because the very low concentration have been found? I mean, is it like in the ppm or what is?
This is a ppm.
ppm.
Yes, low concentration. Well, this is a very difficult question actually, because it is related to the socio-economical issue. This is a hint.
Sure.
You understand? Think about the social, also think about eating habit of the United States. People does not eat so much fish usually, right?
Yeah.
But so you have to, thinking about this background, eating more fish taken directly from the lake means what?
Means more exposure.
Right, exposure, but you have to think about their socio-economical background.
Yeah.
Okay, so again, many people prefer eating meat, beef or pork in the United States, but population around here have to eat fish. Have to eat fish. Why?
Because they don't have money to buy.
That's right.
Right.
That's right. So the population, genetic background, social background is different between this population, that population. Actually, when you relate with smoking, drinking habit, actually, these people smokes more, drinks more. And these people has higher education, higher income. So it's sad to say, but the genetic background could be also different, right? I mean, intellectuality, unfortunately, could be different between this population and this population. So you always have to remember that human being is highly heterogeneous. Okay, so by looking at data, you have to normalize these backgrounds, right? If there is any epidemiologist or public health researcher, you can easily understand this situation. Okay, this was published in 1996, but this researcher did not adjust these issues. So there is a strong protest from some researcher or some company that this effect could be induced by the difference of socioeconomic background or their habits. So this could be the effect of smoking or drinking, not PCB exposure. This is what was happened. Okay,
Yeah.
so this is very difficult.
It's kind of like same I don't know, in kind of a very small district in Indonesia is also same like that, and I mean some of thyroid cases is also increasing in the village area because they close to the sea and they didn't reach to the city so much.
Right.
Yeah, some of kind of this background is also
Mhm.
important to think about.
Yes, yes.
Yeah.
So to solve this problem, you need to clarify the toxicity, right?
Yeah.
So you can do the epidemiological study, such as cohort study, and probably some of you may know about the cohort study. Some of you may not, right? Miski, how about the background? People know about the cohort study?
Yeah, some of them know.
Okay, so you can set up two cohorts,
Mm.
such as people living in the city,
Mhm.
people in the village. So you have to do the two group of cohort, and then you can adjust the age, genetic, how to say, socioeconomic background, or smoking habit, or drinking habit. Probably this is not the case in Indonesia, but it's very big problem in Japan, for example. And then after that, you can compare the difference of like a food, like eating salty food or something like that. So you can exclude bias
Mhm.
by clearly set up the cohort, right? Then you can identify the confounding factor using the appropriate statistics. But then you can identify the effect of environmental chemicals, right? By comparing two cohorts. Understand, right?
Mhm.
Yes. Then, of course, you can measure the EDC levels in blood or serum or whatever. But the problem is mechanism cannot be identified, right?
Yeah. Oh, complicated.
Right. So you have to do the experiment in the laboratory.
Mm.
So you can do animal study.
Mhm.
So thinking about the problem in human, what do you think about the advantage and disadvantage of animal study?
Mhm.
People, can you
Ask someone.
can, don't worry about making mistake. You need to think and answer. What do you think about the advantage and disadvantage of the animal study? You can write 1 or 2 sentences, advantage and disadvantage.
Mm. Okay, anyone?
Don't worry about making mistake. This is the discussion session.
Mm.
If you wish, you can raise your hand.
Yeah, you can just directly on your mic and answer.
What is compared to using human? What is the advantage of use animal study? There are a lot of advantage using animals.
Mhm.
What do you think?
We can know deeply the mechanism, I guess. Okay,
Yeah.
Miss
Yes,
Kiki.
model for research, right? Yes,
Yeah.
model. But how? What is the
So
advantage? Yes.
yeah, so Miss Siti
That,
answer. Yeah, in animal study we can use the animal study as a model for research. Yeah, then how?
yes.
Then how? Of course, we mimic. Okay, who is speaking?
Please identify yourself.
Preclinic study maybe
Yeah,
using mice or rat, and we calculate the lethal dose 50.
Mhm.
Yes, yes, that's good. Yes, dosing. You
Mhm.
can control dose to give. Yes, wonderful.
Mhm.
Any other?
Dosing, and then is there any?
Any other?
So, Miss S, in animal studies, maybe there would be a bias from anthropomorphic views. Miss S.
Mhm.
Yeah,
Yes,
anthropomorphic views.
zebrafish. Yes, you can use zebrafish if you want to.
Mhm.
Yes, you can use a large number of zebrafish, and it's cheaper.
Yeah, and then Miss.
Ethical, yes.
Mhm.
Animal abuse could be a problem, yes.
Mhm.
Clinical procedure, yes. Don't worry too much about clinical procedure. Um, Animal is animal, but still animal is useful to use because we can use you have to use animal
Mhm.
because we cannot use human being to do the experiment. And you can control the dose,
Mhm.
right?
Mhm.
Also, okay. So also on the genetic background is adjustable. Okay, so you can use one specific species, right? And usually we make very specific animals species, mice with completely same background.
Mhm.
Okay, but of course there is a disadvantage. Disadvantage is the mouse is not human, right? So you have to think about the difference between mouse and human. But usually our body system is the same, right? Hormone that control our homeostasis is the same hormone regulate the brain development, but could be the timing could be different.
Mhm.
So animal study have a lot of advantage, right? And the dosing, as you mentioned, timing, you can control the timing to give the drug. You can control dose to give the drug. You can adjust genetic background. You can use only male or only female, right?
Mhm.
And you can do the lot of studies. Like you can do the behavior study, gene expression study, because you can dissect out each organ and check out the protein level expression or protein level, messenger level. So there's a large number of advantages of human using animal.
Mhm.
But disadvantage is still cells are mixed, right? Brain contain different kind of cells. So if you crush the brain, extract the protein, and measure the protein level, still we don't know which neuron is affected, right?
Mhm.
So cell culture is other way to study.
Mhm.
So advantage, advantage mechanism can be clarified, right?
Mhm.
You can pick one specific cells, give the drug, extract the RNA, do the immunocyte chemistry. You can examine the mechanism. What is the disadvantage, for example, on the other hand?
Mhm.
Can you guess the disadvantage of using cell culture?
We cannot the explanation about the metabolism, the whole metabolism in the body, because only
Exactly,
cell culture.
exactly, exactly. Yes, we can examine the we can examine the mechanism of toxicity of each cell, but cell-to-cell communication is different from in vivo. So the whole body metabolism cannot be measured by cell culture, right?
Mhm.
So we have to combine animal study and cell culture study. Okay,
Yeah.
right. So I will show you one model. Okay, so you can think about how to arrange the experiment. So I will show one example. This is Miski's favorite project, gadolinium, but she published a very nice paper. So gadolinium-based contrast agent. Gadolinium is this one, ion. Okay. Gadolinium is very toxic. GD3 plus is very toxic. So you need to curate with organic compound. But gadolinium is very useful for MRI. Okay, so if you give gadolinium, to human being, if you give injection to gadolinium, then brain tumor can be enhanced. You can see an arrowhead. So this is without gadolinium. You cannot detect any tumor by your MRI image. But if you inject gadolinium, you can easily find out these brain tumors because gadolinium can accumulate into these tumors. Okay, but gadolinium are also found in environment because this is secreted in urine. And also, if you repeatedly give gadolinium to human, it cannot be washed out. So even few months or few years after gadolinium administration, some people may have accumulation in brain. So again, fetus and newborn is more vulnerable to environmental stimulation, as we discussed before, right? So if mother or lactating woman are exposed with gadolinium, what's going to happen in fetus or in baby? Nobody knows actually.
Yeah.
Everybody think that it could be risky. So in some country, use of GBCA is generally banned for pregnant women and lactating women. But even in Japan, sometimes gadolinium is allowed use even during pregnancy or even in newborns. Of course, there is risk of new toxicity, right? And some neurological symptoms have been reported in human. But again, human being is very heterogeneous population. So the symptom is not consistent among people. So, okay, so some company came to you asking, please test this compound if it's toxic or not. Okay, some company may come by bringing a new GBCA, right? Or some patient group wants to sue the company because gadolinium was injected during pregnancy, and this woman may have abnormal baby. Then baby so the mother may want to sue the company. Then that mother came to you. I want to sue this company, but we don't know the toxicity, so please test it. Right? This can be happen. So I'll give you 10 minutes.
10 minutes.
10 minutes. I'll give you 10 minutes and think about how you design the experiment.
Uh, maybe animal study, we can use teratogenic experiment.
Teratogenic, yes, this is one. But you have to have more amount for them to cause the cancer. You know, what they want to know is gadolinium toxicity to healthy brain or normal brain. So
Okay. Teratogenic is the transfer from the mother to the offspring, right? So maybe the effect, how maybe Mr. Suwendar means to how the gadolinium may transfer to the placenta and directly goes to the baby.
Yeah. Yeah, that's, yes. This is one answer, right? Animal study. Any other? Think about it, and we have time to answer.
That's my study. I'm kind of missed this study actually. I haven't finished to write the paper for you, right? For this one. Yeah.
I'm waiting for you, your paper. Actually, she's on the way of conducting animal study, and she was supposed to write one more paper. Actually, even now,
yeah,
I'm waiting. You know, you have all data to write a paper.
yes.
Study, but you haven't written yet.
Yeah. On the way.
Yeah, I can introduce her data anyway.
Yeah.
So what you want to know? Okay, so what you want to know is the effect of gadolinium in developing brain, right? This is the question. Research question is effect of gadolinium in developing brain. So to find out the effect in developing brain, what kind of experiment you can conduct? One, again, even if you use a newborn, you may not be able to detect abnormality because accumulation of gadolinium in brain is not so high. And such gadolinium may disrupt hormone-mediated brain development or may directly affect to the developing brain, but not too strongly. So even if you use a newborn, you may not be able to detect it. So what kind of animal you have to use and how you give gadolinium? This is a very difficult question, but you have to think about it, you know, because you have to conduct, particularly for young students, you have to design experiment by yourself. So clear, you have to find research question. Okay. 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 Can I use the whiteboard, Miski?
Sure. So you have, yeah.
Okay. Okay. So research question. Okay.
How?
Toxicity, right? Oops, sorry. And during development or development exposure. Right? And outcome. Right? So you want to study the gadolinium toxicity, and you want to examine the development exposure, effective development exposure, and the outcome. These are studies that you want to clarify, right?
Yeah.
So by examining this study, what kind of experiment you need to design? Okay. So experiment. Let's say, for example, let's say mouse or out. Oops.
Mm.
Anything, either is fine. Okay. Gadolinium injection.
Mhm. So maybe people will ask, is it okay if
Yep.
we give the gadolinium orally
Oh, that's
in,
fine. Yeah,
in the experiment? Do you think is it fine?
I believe so. Because some gadolinium may be already ingested through breast milk.
Uh-huh,
Okay.
uh-huh.
Right?
Yeah.
That's what you are doing.
Yeah, because previously I was like thinking if we want to expose animals with gadolinium, we need to make as similar as we can. I mean,
Yeah.
similar to the human cases, right?
Yeah.
Then it's really impossible if human actually adult can get gadolinium by oral unless the gadolinium is contained in the water or something like
Yeah,
that.
this could
Right?
be also because some river is contaminated by gadolinium, right?
Yeah.
So this is also possible. So you can use
Mhm.
gadolinium injection or gadolinium ingestion at certain period of time.
Mhm.
Then
Dose.
dose, right?
Yeah.
Different dose. Or equivalent human, right?
Yeah.
Comparable dose to human.
Mhm. Mhm.
So you can give the gadolinium. Then outcome.
Evaluation. Yeah.
Outcome.
Oh yeah.
How do you assess the outcome?
Maybe pharmacological profile.
What do you mean?
We can observe the effect
Yes.
to neuron system,
Yes.
to locomotoric system, digestive system, and so on. So we can make a conclusion.
Okay, good. Yes.
Yes.
Very nice. Yes. Neurological symptoms. How do,
So Mr. Yuda also say that animal study is still less
yeah, yeah.
applicable and predictable.
Yeah, your
Okay.
symptom by applying applying sorry. In like a locomotion, right?
Yes.
Applying test. Right, but locomotion test, etc. Okay, so actually this is what Miski did when she was in Japan. So we discussed and studied. So this is what Miski did. So she injected gadolinium during pregnancy and after birth from E15 to P19 in mice. So to measure the gadolinium level, she picked up the brain at postnatal day 28, which is almost about the period for puberty. After that, when mouse became adult, she tested several different behavior. I do not have enough time to introduce all of her data, but she did some behavior analysis. So this is the concentration of gadolinium, and she used different kind of gadolinium, but you have to know that certain level, certain amount of gadolinium is contaminated in brain. And macrocyclic gadolinium seems to be more safer than the linear gadolinium. And compared to male and the female, I don't know, but the female brain may contain more gadolinium than male. And the gadolinium was retained throughout the brain in both DAM and PAPs. And in PAPs, the total gadolinium concentration was higher in the gadolinium-treated group, of course. Sorry, linear gadolinium. This is linear gadolinium. And the gadolinium concentration is higher in female in both groups. I don't know why. We still don't know why, but there may be some crosstalk between estrogen and gadolinium, which allow them to accumulate more in brain. But still, we cannot find out the estrogen-like effect of gadolinium so far. But at least the gadolinium was accumulated. Also, what she did is she examined the memory and the memory in cerebellum, hippocampus and the cerebellum. Also, she measured the motor coordination by using rotor rod. I would like to skip the open field test today. This is to examine the anxiety, but difference was not so great, so I'd like to skip this. So this rotor rod, you can put the mouse on the rotating rod. So mouse hates height. So mice try to walk on this rotating rod, but if their motor coordination is not so good, then mice easily falls. And on the other hand, this is to examine the memory. Okay, she tested two different kinds of memory. For example, this is object recognition memory. You can put the mice into one box. That you can put two same object. Mouse were very anxious what these are. So mouse approaches and poke their nose and smells at the same level to these objects. Then after that, you can remove the mice, let it sit for 10 minutes. During that period, you can replace one object to a new object. Then if mice remember the previous object, this object is pretty new to mice. So these mice can approach more frequently to this new object, right? So by doing so, you can test the mice memory, of recognition of new object. The other apparatus that she used is object in location. So again, you can put the same object and put the mice here. Then you can remove the mice, and 10 minutes later, you can put the mice again, but you can move this object to the new location. So if mice remember the previous location, then this location is pretty new to mice. So mice try to approach more to this object, right? So this is what we call object in location. So important thing is you need not use sophisticated material, okay? You can do the same experiment even in Indonesia, okay? You can just, maybe it's better to have a videotape. Only you use a box, an object. Maybe you can have a video camera, or even your cell phone is fine. and stopwatch. That's all.
Yeah, I will
Right?
make it. I will make it in this few
Yeah.
months. I say.
Yeah. So we do not use a very specific sophisticated material, but Miski did excellent work using this. And also this open field rotor rod apparatus, again, is not so difficult to make. You can just prepare the rotating rod with certain speed, right? And you can prepare the guard or barrier among mice. Then you can measure the cerebellar function or motor coordination function. And this is to test hippocampus. And this is to test perirenal cortex. Am I right, Miski? Right? This
Yep.
is hippocampus, this is
Hippocampus.
perirenal cortex, right?
Yes.
Okay. So this is to test the memory related to cerebrum. This is to test the memory related to hippocampus. This is to examine the cerebellar function. And what she found is in rotor rod, this is control. And if the mice is exposed to the linear gadolinium, time to fall to here is decreased. And actually a female, you remember that female had higher accumulation of gadolinium, and the female effect is greater than male. So exposure to gadolinium can produce abnormal cerebellar development, but just by watching the mouse daily behavior, you cannot detect it, right? You need to have some special test to study the effect. Also, this is object recognition test. This is OLTS. This is, I think, this is object recognition test. So you can change the object to new object. And again, this linear gadolinium exposed mice cannot discriminate the object. So this is the ratio to approach the new object. The object, the discrimination is 50%. It means this mouse approach all the object and new object at the almost at the same time period, right? Object location also, when mice are exposed to linear gadolinium, this mice cannot recognize the new location. So by doing so, you can examine memory acquisition. Oops. Further, Miski also did other experiment because she wants to know the effect during lactation. So she just give mice gadolinium injection to the lactating mice from P5 to postnatal day 5 to postnatal 9. Okay, so pups were exposed to gadolinium through breast milk. And I'm not gonna have enough time to introduce. So this is what Miski did. So she's very cruel. She tried to suck the milk from from mother. Right? Look at that. Well, I'm just joking. Sorry, Miski. But she successfully took out the milk from mother, as you see. So we measured the gadolinium concentration, and actually it is very high in milk. And she examined the behavior.
That's
This is Miski took this video actually.
yeah, it's
Right?
pumping.
It's pumping. So she's taking the milk by pumping. Anyway, so even with the through the breast milk, you can see open field and sorry, rotarod and the object cognition test and looking test. All of these are disrupted by lactating exposure of the lactation exposure to gadolinium. So she did excellent job, and we clearly have shown that gadolinium exposure can disrupt brain development even if at a low dose. Okay, so particularly, sorry, this linear gadolinium have very strong effect onto the macrocyclic gadolinium. I have to warn that recently in Japan, this gadolinium was banned to use for very few, except for special occasion. We are not allowed to this gadolinium because of toxicity. And we are only allowed to use this gadolinium recently in Japan. But I have heard from Miski that this linear gadolinium is still allowed to use in Indonesia, right, Miski?
Yeah, it's cheaper. So
Yeah.
yeah, it's some of hospital actually still provided tooth. Actually, yes, if the patient can pay more for the macrocyclic one, of course the medical doctor will give the macrocyclic. But unless if they use the there's no money, so they use the linear one because it's cheaper.
But I strongly suggest people who is pregnant or who has lactating, do not use linear gadolinium from your study,
Yep.
right?
Yeah.
Okay, we do not have enough time.
Yeah, it's a 15
So
minutes left.
yeah, only 20 minutes.
Yeah.
So I would like to introduce the cellular study also. Then after that, we have maybe a few, 10 minutes of the question time. Okay, I would like to introduce also for, okay, for cellular study, this was done also by the Indonesian, okay, Winda Ariani, who is from the, but she's not from originally from Bandung, right? She's from
It's from
Yogyakarta.
no, Surakarta.
Ah.
I forgot.
But she used to work in the Bandung Institute of Technology, right?
Yeah.
Right, and she also she was also in the PhD student on my department, and she mainly conducted cellular study, and also she also published a very nice paper regarding the gadolinium. So what she did was she used cell, and she measured the transcription of thyroid hormone receptor mediated. She measured the thyroid hormone receptor mediated transcription. So look at this. If you give the cell to thyroid hormone, you can measure transcription. And if you give thyroid hormone and gadolinium, in some case with lower dose, thyroid hormone mediated transcription was increased. But by increasing the dose, thyroid hormone mediated transcription is suppressed by gadolinium compared to this. This effect is only seen in linear gadolinium, okay? And cyclic gadolinium, you don't see any effect. And gadolinium chloride, this is very, very toxic. So you can just see the cell death. Anyway, I don't forget about this result. You can just compare this and this. And also she gave the gadolinium to neurons, in culture. So if you give thyroid hormone only, you can induce dendrite growth. You can compare this and this. If you give linear gadolinium as for the result of transcription data, you can see the very increase in dendrite arborization, dendrite growth. Okay, but with the increase in dose, again, dendrite growth is inhibited. So its result is consistent with transcription data. And even if you see the increase in dendrite growth, it does not always mean neuron grow faster or neuron grow better, because abnormal growth, abnormal rapid growth of neuron may also disrupt our neuronal function. Okay. And oops, sorry. And she also did examine the neuronal migration. So she cultured the cell onto the plate and made a scratch of the cell to make wound like this. So you can get rid of the cell here to make artificial wound onto the plate. Then if you let it sit for 24 hours, cells start to migrate to heal the wound, okay, like this. So if you give several different kind of gadolinium, this process is, I don't know why, but this process is activated. So cells start to migrate faster. So this is a cell-free area after 24 hours after making a scratch. So decreasing cell-free area means migration is more active in this with this compound. So I don't know why, and we still don't know the physiological significance of this phenomenon, but gadolinium can accelerate the cell migration in culture. And also, gadolinium can induce actin formation around the cells, because this is control, and you can see blue is a cell nucleus. So you can see the network of actin around the cell. And this kind of network can accelerate the cellular migration, by making the philopodia. So together with increasing cellular migration and actin, cellular actin, actin in the cytoskeletal protein, actin formation is also activated by gadolinium. And this RGDFB is an inhibitor of membrane thyroid hormone receptor. Thyroid hormone also have a receptor in membrane. And if you give the thyroid hormone receptor antagonist, this kind of increase was suppressed, indicating that thyroid hormone could be involved in this kind of phenomenon. Okay. So this is the summary of the gadolinium study. So gadolinium can disrupt thyroid hormone action, neuronal cell growth, sensory. I did not show you, but sensory threshold, pain sensitivity is also disrupted by gadolinium exposure. Memory acquisition, motor coordination, and neuron glia migration. So these are all disrupted by exposure to gadolinium. So appropriate compound should be used and avoid using for pregnant women. And also you have to inhibit contamination of the river and soil from gadolinium dumping. Okay, so this is all for today. So we still have about 10 minutes for discussion questions. So if you have questions, please give me. Oops.
Okay. So there's one question from Miss Dina Sensei. Dear Professor Koibuchi, thank you for a very comprehensive explanation. I have a question related to the effect of PCB. How far genetic background could give an impact? What features of the gene that might be sensitive to it? Is it possible to be treated through a gene therapy, for example, or it would have been too late?
Okay. Genetic background. Well, like you can think about it. Some people can easily become diabetes. Other people may not, even if you become obese or eat so much food. So this is because genetic background is different, right?
Mm-hmm.
So such things can be applied for the sensitivity to environmental chemicals. Some people may be sensitive or vulnerable to EDC exposure. Some may not. So genetic background affects the severity of the environmental chemicals. Again, because we have such kind of problem in human, doing experiment using human is very difficult, and assessing endpoint of EDC exposure cannot, well, I don't say cannot, but it's very difficult to identify, right? Because somebody may have a phenotype, somebody may not. That's why we use animal for endocrine disrupting chemical research. And the gene therapy, it is very difficult because once you become adult, gene expression becomes very stable, and it is very difficult to alter, or it is very difficult to induce development or differentiation of neurons after adult. I would not say it's completely impossible, but I would say it's very difficult. But once IPS technology is fully developed, we may be able to produce new neurons, and that can be injected in brain and produce new neuronal circuit. But it is very difficult, I would say.
So, Miss Dina, do you want to talk directly to Professor Koibuchi?
Yes, thank you, Professor. So I just thought maybe if we learned about pharmacogenomic deeply, maybe we can see what gene that get an effect from the PCB, and then maybe with the technology of CRISPR-Cas or something, maybe we can do prevent for for some ethnic to more harm about PCB.
Yes, this is what we are aiming at.
Yeah.
Okay. I did not show the result of gene expression, but of course, we measured the expression of so many genes,
Yeah.
And sometimes we use a microarray. You have heard about the microarray, right?
Yes.
So, or next generation also, and also we sometimes apply next generation sequencing to examine the mechanism. So we have identified several genes responsible for such kind of phenotypic changes. So by measuring the expression level of these genes, we may be able to find out the target gene. So by identifying the target gene, we may be able to predict the abnormal phenotype of human being, right? So at least we can diagnose correctly whether some phenomenon or some symptom of human can be induced by the toxicity of the certain chemical, right? So, so this can be a very nice indicator of prevention. So if you find out the early symptoms, then we can prevent from progressing, right? And if we can identify the genes of interest, target gene, we may be able to prevent the adverse effect to such genes, right, by giving some chemicals. So this is what we are aiming at right now. Thank you very much for your comment.
Thank you, Professor.
Yeah.
Thank you, Ms. Dina. Okay, ladies and gentlemen, is there any question before we close this session? Okay, Ms. Sri Sumartiningsi, you raise hand. Would you ask directly to Professor Koibuchi? Sure. Halo. Ibu Sri Sumartiningsi ada? Silakan Ibu bertanya langsung. Alright. Mangga, Ibu. Bu Sri? Ada? Tidak terdengar suaranya? Oke. Bu. Oh, maybe something happened. Okay. Okay, maybe today's, oh, okay.
Wow.
Wow, it's kind of,
Wow.
Hello.
Hi, Professor Adrianta. How are you?
Fine.
wow. You know each other.
Yeah, I like to ask you a question
Yes.
about the gene and the environment. So, can an environment affect the gene expression, or can environment directly cause changes in hormone or something like that, not via gene expression? Do you understand?
Yes. I think regarding the brain, you have to have some stimulation through through neurons or hormones. So environmental chemicals can affect neurons, right? And
Directly,
it can,
yes.
yeah, directly, and it can
Yes.
mimic like hormone. So you have to have hormone receptors, right, to
Yeah.
induce environmental chemical action. So, but and such kind of action can change the gene expression. So it is not that, so environmental chemical does not act directly to the DNA.
Yeah.
In many cases, but it changes the transcription. And but in some case, gene or DNA can be, how to say, disrupted.
Yeah, of course.
Some hormones, some environmental chemical can make the wound of DNA, but this is quite rare. In many cases, environmental chemicals can alter their messenger RNA level or transcription or whatever. So it's mostly like epigenetic mechanism.
So it's epigenetic?
Yeah.
Okay, epigenetic.
In many cases,
yes.
In many cases, yes, I think so. Yes.
But like radiation of a bomb.
Mm-hmm. Oh, it's a different story.
Directly affect the human, not via the genes, maybe, isn't it?
Well, atomic bomb or radiation can disrupt the structure of the DNA. Radiation can easily cut out or disrupt the DNA bond.
Yeah.
Then during the repairing process, some unexpected thing can be happened and causing cancer. Or some if the repairment can be induced, then people will die.
Yeah. But that is not gene expression, isn't it?
No, DNA itself. Yeah.
Yeah, DNA directly itself. Yeah.
Yeah.
Okay. Second question is,
yes,
the human body must have some defense for your disruptive agents. What are the defense mechanism of our human body to get rid of those toxins, like from the fish or something like that?
Yeah.
You said.
Yes. Naturally, we have a system to catalyze drugs
Yes.
which is located in liver, which what we call cytochrome P450 enzymes.
Yeah. Yeah.
Can catalyze some drugs or chemicals. But unfortunately, it is quite recently when we start to be exposed to the environmental chemicals.
Mhm.
Even if we have a cytochrome P450, the ability to break down or catalyze or excrete environmental chemicals is limited. That's why environmental chemical can be easily accumulated in brain. But hopefully by accelerating the cytochrome P450 action, we may be able to get rid of such kind of chemical, but still we need to do more research on that.
Yeah. You said fish is consumed a lot in Japan, not in Europe, not in America. Indonesia, maybe fish is only on the seashore. People living in the seashore, people in the mountain never eat fish too far from the sea. So fish has omega-3s.
Right.
Omega or
yeah.
The good omega.
Yeah.
Can it help to destroy your toxins?
I don't think omega-3 fatty acid directly contribute to get rid of the toxin, but omega-3 fatty acid is very helpful for your body. That's why in Japan we encourage people to eat more fish. But of course, again, there's a risk of environmental chemical exposure, right, from the fish.
Yeah.
So yes, we are always discussing about such kind of possibility. And in United States, federal government prohibit of eating tuna limited to the twice a week or something like that because they worry about the mercury contamination of the tuna meat. But we do not do that, because we measure the mercury in tuna and the level will be very small. Anyway, yes, this is also a good point to discuss. Yes. Thank you very much for your comment.
Thank you.
Thank you.
Prof. Nori.
Yeah.
Bye-bye.
Bye.
Thank you very much for Professor Adrian and all the participants. It's a really nice talk from Professor Koibuchi. Thank you for coming to us, Sensei. It's really nice
It's
time.
nice Yeah.
And we hope to have you again next time.
Sure.
Okay. So, Sensei, before we close this session, let's take pictures together.
Sure. Like this.
Thank you very much, Professor Koibuchi.
Yep.
Yeah. Okay, everyone, please turn on your camera. I will start the session. So the slide 1, 1, 2, 3, smile. Okay. Slide 2, 1, 2, 3. Slide 3, 1, 2, 3. There are 5. 1, 2, 3. Okay. 1, 2, 3. All right. Finish, Sensei.
Okay.
All right, everyone. Thank you very much for coming to this session. I'm Miski. Sorry, I forgot to introduce myself. Oh my gosh. Sorry. Thank you very much for coming and participating in this event. And thank you, Koibuchi Sensei, and everyone. If anyone wants to keep contact with Koibuchi Sensei, maybe is it able to send you email sometime?
Yeah.
Sure. Okay. From Department of Pharmacy, Universitas Islam Bandung, I would like to say thank you to all and thank you very much. Alhamdulillahirrahmanirrahim. Wassalamualaikum warahmatullahi wabarakatuh.